Well, ahoy one and ahoy all. Today we're going to be talking about wort sugar utilization during a 9-generation serial repitching experiment. So hopefully throughout this presentation I'll be able to describe exactly what that title means. So just to get everybody oriented, I want to talk about the brewing process itself. It only consists of 4 ingredients, being malt, water, hops, and yeast. We first begin with the malt in the milling and malting process and apply it to a masher with water. So mixing this together, we get our mash, and then from there we can apply it to our boiler where we add hops. And once we remove all solids from that, we have our wort. And our wort is a liquid substance that has all the carbohydrates that yeast need in order to break down and actually have the fermentation process take place. After that, we can remove the yeast and we can have the maturing and packaging process. But for the purposes of this presentation, we're going to focus exclusively on the fermentation process. So, a couple of terms I need people to understand is first serial repitching. And serial repitching is the process of re-inoculating a previously used yeast culture. So that sounds nice, but what's that actually mean? We have generation 1 of yeast, and that's your brand new, right out of the bag, freshest yeast you can get, ready to go. You can apply that to the fermentor with your wort, and you get your fermentation, and this is your first fermentation. What serial repitching is the removing of that yeast culture you just used and calling that your second generation of yeast, and use it again in another fermentor with another fresh wort. And you can keep this process going indefinitely, hypothetically, but most large breweries tend to use it about 8 to 15 times, and it really varies on the amount of times you can use it due to the health and viability of the yeast cells. Additionally, there are only 5 major sugars utilized by Saccharomyces cerevisiae, which is the common ale yeast, which is the yeast we used. Specifically for our experimentation, we used a German ale yeast strain. So as we can see right here, we have fructose, sucrose, glucose, maltose, and maltotriose. And the reason I have the 3 sugars on the bottom in a red rectangle is because these are known as the major wort sugars. These sugars are in the greatest concentration in the fresh wort and create the majority of the fermentation capabilities of the wort. As we continue, glucose is a monomer, and maltose are 2 glucoses together, with maltotriose being 3 glucoses together, and we can have this thing going on indefinitely, hypothetically. And once we get past this point, our first one being maltotetraose, we call these dextrins, and dextrins is really a colloquial brewer's term for unfermentable sugars. And the first one is maltotetraose, and that's 4 glucoses binded together. And it's always been determined and claimed that these dextrins are unfermentable because they are too big for the yeast to actually utilize. So the purpose of our experimentation was 2-pronged. What we first wanted to do was validate previously reported claims stating that the pH drop, gravity drop, as well as the utilization of the major wort sugars remains unchanged throughout all serial repitches. Additionally, we wanted to build our own novel claim by studying maltotetraose and seeing if this sugar is affected at all by serial repitching. In other words, is maltotetraose utilized through induction of serial repitching over time? So first we can see our starting pH and ending pH, the standard pH drop. over the fermentation process, and we see that it does not change over time. Very consistent and very stable. Again, the starting gravity as well as the ending gravity are gravity drop. There's no major change over these 9 serial repitches, which is something that's already reported and we're just validating. Before we continue for the utilization portion, first wanted to go over our methodology of HPLC, or high performance liquid chromatography. And so this is a way of separating analytes through a high pressure column and discriminating its constituents by affinity to the column. So what this looks like is we will load our sample and our solvent of water as we push it through in high pressure. Sugars that have a strong affinity to the column will move at a slower rate than sugars that have a weaker affinity to the column. And we can put this on a chromatogram over time and have a result like this. So this is our HPLC trace sample of our pre-fermentation wort, our wort before any yeast has been added. And we can clearly see our glucose, maltose, and maltotetraose peaks, our major wort sugars, and all these other ones down here are simply our dextrins. And for our generation 1 post-fermentation wort, we see exactly what we would expect. The major wort sugars have been completely utilized with the dextrins remaining untouched. And we see this through all 9 serial repitches, generations 1 through 9. Glucose, maltose, and maltotetraose, the utilization of them remains very stable and very consistent throughout the entire experiment. Now we're going to turn our attention to the dextrin peak maltotetraose. So once again, this is the 4th peak, and it is 4 glucoses binding together, the first dextrin, first unfermentable sugar. And what grabbed our attention is during generation 3, comparing the pre-fermentation and the post-fermentation wort, we saw a major reduction in the maltotetraose peak. So this is what really sparked off our experimentation. We wanted to see if this holds true throughout all other generations and how significant of a decline or utilization is it. So as we see in generation 1, we see exactly what we would expect. There is no reduction in maltotetraose, a very horizontal trend, beginning at 100% and ending at 100%. We saw this in generation 2, but when we get to generation 3, this is where we see our major utilization taking place, around 95% utilization of this maltotetraose sugar peak. We also see this in generation 4, and in this line right here, generation 5+. This is just an aggregate of generations 5 through 9. And we still see the same trend taking place, a major reduction and utilization of this sugar. And this table right here does a good job of summarizing our results of this peak. Generations 1 and 2, there is no utilization. But generations 3 to 9, we see a major reduction or utilization of this maltotetraose sugar, from its highest being at 95.7% utilization to around 87.1% utilization, but a very significant decline in the sugar peak, showing major utilization, a previously unreported and novel claim we are making. So a big question that came up was, what type of enzyme is being represented here? Is it intracellular, maybe a transport enzyme? What about extracellular? To test this, we took the supernatant fluid from several of our samples from generations 3 and 4 and supplied it to a fresh wort. So if this was a transport or intracellular inside the cell enzyme, we would see no reduction in these peaks because the yeast has been completely removed. But if there's an extracellular enzyme, then it would still be present in the samples or the supernatant fluid. Unfortunately, due to the pandemic, I was unable to pull off the charts from the computer in order to make this presentation. So we're just going to have to go off you trusting me of what I saw before spring break. So, so what we saw, at least what I saw, trust me on this one, saw a major decrease in maltotetraose and a major increase in glucose. So what does this mean? What does this mean in this picture I can't show you, but you're just trusting me on? What this would mean is that the protein is not a transport enzyme or an intracellular enzyme. What enzyme is being represented is an extracellular amylase or dextrinase. And what's very exciting is it only works on maltotetraose, only works on N equals 4 glucoses binding together. And the way it does it is it breaks down maltotetraose into glucose monomers, which can be very easily transported across the yeast cell membrane for utilization. So a very exciting enzyme that we've discovered and follow upon here, and very novel and unreported. So this is very exciting for us. So to wrap this all up, what do we get from this? So serial repitching does not influence pH drop, gravity drop, or the metabolism of major wort sugars. And this has been previously reported and published many a time. The novel claim we're making is that serial repitching does influence the metabolism of maltotetraose. So some people may be thinking, like, well, is this a microbial contamination that we're seeing right here? If this is so novel and unreported? Well, we have two reasons to push against this idea of a microbial or some sort of diacetycus or STA1-like contamination. First is the simple idea of the health and viability of our cells. Throughout all 9 serial repitches, throughout all 3 fermentors, we kept a yeast cell count, standard methylene blue cell count through ASBC standards, and our cells were still healthy. Additionally, the standard amount of pH, gravity drop, and metabolism of the wort sugars adds to the claim that our cells were still very viable throughout all 9 generations. The other thing I want to point out is the difference between specific versus non-specific enzymes. What we're seeing here is a very specific enzyme in that it only acts on maltotetraose. If this was a microbial contamination, we would be seeing a more non-specific enzyme being represented. And what would that look like? What that would look like would be looking at the post-fermentation wort. Instead of having any other dextrins being represented, we would be seeing just a simple flat line because these non-specific enzymes act on all sugars. So there would be no dextrins still present. All sugars would be taken down, and that's not what we're seeing. We're seeing a very specific enzyme acting specifically on maltotetraose. So what? What's this really mean? The first question we really had is, remember, we only worked on German ale strain. What we're wondering is what about other strains? There are thousands of other strains out there, and how does serial repitching affect the utilization of dextrins on those specific strains? Can it go farther than maltotetraose? Can it break down an N equals 5 or N equals 6? Or how many other sugars can they metabolize? And what about increased amount of serial repitching, such as using 15 generations? These are questions we hope that others will explore along with us. This new field of looking at serial repitching and the intersection of dextrins. So this would have an effect on the mouthfeel of the beer as well as the fermentation process itself. So to close everything out, I first want to give out some Acknowledgements to my PI, Dr. Will Deutschman, for giving me the opportunity to explore this experiment throughout the majority of my college career, as well as my team members, Bobby Brueggemann, Mary Grace Lewis, and Sam Dolsky, for supporting me throughout this entire process. Uinta Brewing, who was our who was our only supply of fresh wort throughout our entire experimentation, and Isaac Winter, who was our main source of contact at Uinta Brewing. And of course, we would like to thank the American Society of Brewing Chemists, for we have presented our research at their conference, last national conference, and we were prompted to submit for publication. So we are currently in the editing process for that. So thank you for listening, and if anybody has any questions, just leave it in the comment section below, and I will do my best to try and answer them. So thank you for watching.